that's another give us an idea of the presence of this invader on that plant. And sometimes I just want to point out that sometimes when the larvae are very big and have been eating a lot that leaf, we sometimes don't see that zigzag pattern that clear anymore. So it might be a little bit harder to identify the presence of the sawfly in this case. And I also want to mention that, like, just for comparison purposes, that there might be another patterns that we might find on leaf. Like, for example, this one left behind by leaf miners. And I just want to point some differences so we are able to identify this sawfly properly. In the case of the leaf miner, the damage on the leaf are going to be kind of superficial. It's not going to go through the whole leaf, which is a difference in here that we see that the larvae is going to eat all across the leaf. So once that we know that we are finding that, we can report it, we can take measurements. Another thing that I want to mention is that this species is a very successful invader. This species reproduce parthenogenetically, and that means that it is just a form of a sexual reproduction in which we can have embryos that can fully develop from unfertilized eggs. And that means that females can produce offspring without even mating. That eliminates the time of the sawfly finding a mate in order to lay the eggs. In this case, they just can lay the eggs without mating. Another thing is that the eggs would only produce female sawfly. There are no reports of males sawflies up to this day. So imagine this species can lay 60 eggs, and all those 60 eggs are going to be females that are going to be ready to lay eggs as soon as they emerge from the pupa, without needing the need of finding a mate or reproduce at all, or copulate at all. Sorry. So another thing that is helping them to reproduce faster is that they can produce up to 4 generations per year. So that means that they can go through the whole life cycle 4 times a year, and that's in the wild. There are some laboratory experiments in which they found that the species can have up to 7 generations per year, although in the wild only 4 generations have been reported so far. And another thing that made them a very strong invader is that they are very good flying around. They can move up to 90 kilometers per year, around 56 miles. And that also helped them to disperse and spread out very easily and faster. So taking into consideration all these factors that I just mentioned, when we first found about the introduction of these species in North America, the detection in Quebec, and how it have been spreading across the landscape in North America, we wanted to have an idea of which areas were suitable for the establishment of these species in this new invaded region. So this is where I'm going to mention a little bit about the research that I and some colleagues that I'm showing here on the screen did together. This research was done back in 2024, and we submitted the results as a preprint in 2025, and now we have the results being considered for publication in Integrative Zoology. So I'm going to show you, first of all, some of the basic behind it. The objective of this research was to estimate area of potential distribution of the elm zigzag sawfly in North America, but also have an idea of how this species can have an impact on the elm species in this region. And for doing that, we first go back to the theory of species distribution and base our study there. What are the conditions that determine the distribution of a species in an area? So Sovereign and Peterson in 2005 mentioned that for an area to be suitable for a species to be distributed, we need to have 3 main factors there. We have to have the right biotic interaction, the host plant, no competitors or predators, the right prey. All those conditions have to be in those regions, but also we need to have the right biotic interaction, the biotic condition. And that means the environmental conditions, the right values of temperature, precipitation, humidity, all those values have to be there. But also the species have to have access to those locations in order to be present there. And that's the mobility portion of this diagram over here. So only in those areas with all those three factors overlap, which is represented here in the middle, is when we are going to have the species present there. We can, with some tools, characterize some of these conditions. For example, the abiotic conditions can be characterized using ecological niche models. How these models works? Well, we do have, let's say that we have occurrence of a species, like in this case is Vespa mandarinia, another invasive species in North America that got eradicated from this region. But this species is native from Eastern Asia as well. And here in the map, we can see in red all the occurrences in the native range of this species. We know that the species is there, right? We know that they are living there right now, and we can characterize the environmental conditions in those places and have a reconstruction or that environmental niche that the species like. And once we have that reconstruction of the environmental conditions that the species like because they are using it already, we can see and project in other areas and look in other areas that might have the same or similar environmental conditions to those of their native range. And this is what we can have as a result. We can have a prediction of highlighting the areas that might have the same environmental conditions that are suitable for the species. Now, why are we able to model those distributions? Well, if we look at here in this figure, the left portion is representing the Americas. And we are seeing in gray all the Americas and a little square in blue. If we map all the combinations of temperature and precipitation that are represented in that geographic region in this map on the right, we see here in gray all the values of temperature and precipitation in all the Americas. But we can see here in blue all the combinations of temperature and precipitation of this specific square that we have here, the geography. So we can have that idea. But also we can put it back. We know if we have some combinations of precipitation and temperature, we can see which areas in the map, in the geography, might have those same values of precipitation and temperature. And those are the basics of the ecological niche modeling framework. So also I want to mention that these distributional ecology questions integrates very well with the invasion ecology. Because if we have the invasion curve represented here, like in the x-axis where we have time of invasion, the y-axis we have area affected, and we have different measurements for preventing the invasion, eradication, containment, and long-term management. For the distributional ecology perspective, we can ask these questions. Where my invasion happens? Where are invasions happening? Where can invasion span and how that expansion is going to occur? And all those questions are going to be responding to different sections or are going to be useful at different sections of this invasion curve. So based on all this theory that I just mentioned, we tackle the questions of the soft line. For tackling, we developed ecological niche modeling. And first of all, we needed species occurrence data that we gathered from the literature and museums, private collections. And then we cleaned that data. We perform different steps of cleaning it in order to reduce the bias that are associated with all those type of data. Once we have those occurrence data, we also need environmental data. in order to characterize those places where the species have been already reported. And for that, we use environmental variables at 5 kilometers of resolution, and we compile all that information into principal components, and in order to avoid environmental variability, environmental collinearity, and correlation of the variables. And once we have those two types of data, we already have occurrence and the principal components as environmental factors, we develop the models. The models we did it on Maxen. We performed calibration steps, evaluations, and selected the final models. I'm not going to go into a lot of the details of this section, but I will be happy to answer questions about it later if you have questions about it. And once we have that, we obtain a prediction. I'm going to show you first how we calibrated the model a little bit. For calibrating the model, what we did was to use occurrence from two different regions. First, we calibrated the models only on the native area over here that had all the occurrences reported in Eastern Asia, and projected the models to North America. And in another step, we also calibrated the models with all the native occurrences, but also the invaded ranges in Europe, just to see which option predicted better the current North America distribution and the known current distribution in that region. We did not use any of the known occurrences in North America for calibrating the models, and we left those occurrences only for validating the results that we obtained afterwards. I'm just going to make that note. So some of the results that we got, like when we calibrated the model on the native range, represented here on the right bottom corner, this is the result that we got. Here in purple, I am representing the potential suitable area that we found for this species in North America. We can see the distribution over here and a little bit on the western part of the United States, Canada as well. And if we compare those predictions that we got with the current known distribution of the species in North America, we found out immediately that the models were under-predicting. They were not performing as well because there were a lot of places in which we know that the species is actually present that our models were not predicting as suitable. I also going to mention again that these points here are from the distribution back in 2024. We have now a more like an updated map from Otten and collaborators in 2025, showing how the species have been expanding their distribution. And I have it over here. But even with this updated distribution that we have here, we can see that this model is not predicting very well, only considering the native range for calibrating the models. So we try, as I mentioned, calibrating the models with the native range, but also including the occurrence in the invaded areas in Europe. And when we did that, this was the result that we got. The predictions that we got are more broad, and also they include all the areas where the species have been actually reported already. And also, this was like, as I mentioned, back with the occurrences back in 2024. But if we look at the ones 2025, we can see that our models were able to even predict the distributions in North Carolina, in Tennessee, which is a very more recent detection of the invasive species right there. So we were very happy to see these results, and we were confident that these results were more informative than the one that we had before. And this highlight the example that for invasive species, the point of invasion matters, because in the case of the North American invasion, is now believed that invasion is coming from Europe. There is a very preliminary molecular work done, and it looks like the invaded species in North America is closely related to those in Europe than what they are to the native range in Eastern Asia. So at the moment of modeling invasive species, the point of invasion matters a lot at the moment of detecting uh, and including larger areas of suitability and detecting and getting a better result and a better prediction. As I mentioned before, this is only the sawfly, but I mentioned before that we also included almost species. And here are all the occurrences that we use for creating the models of almost in North America. And in this case, by including the almost distribution, we are not only going to take into consideration these abiotic conditions that I have mentioned before, but we also going to include some of the biotic factors because the almost is the host plant, the host plant of this species as well. So with considering those results, this is what we got. In purple is the sawfly only. In light green, we have predictions of suitable areas for almost species only. And in dark green, we are representing both of them with both suitable areas overlap. And again, I'm going to show you the distribution of the different states that the species have been reported already. But we also can see that we have other places like Arkansas or Kansas and a little bit of Oklahoma that are also in risk and the species have not been reported before. Look at Missouri as well. So we do have a lot of places that where the species have not made it there yet, but they have suitable environmental conditions that might be relevant for the species. And also they do have the host plant as well in those areas. I also wanted to show you an exploration of how the environments of these different locations look like. And here I'm going to show you, these are the maps that I showed you before here on the corner. And on the top, I'm showing you the calibration area in Eastern Asia, sorry, in green, in the invaded area in Europe in blue. And I'm dividing North America representing only the central eastern United States and the southeastern United States in pink. And if we represent the environments in here, representing in these areas in two principal components, principal component one in the x-axis and principal component two on the y-axis, we can have this cloud of points. Each color represent a region. And we can have Eastern Asia environments represented here. Here we have Central Eastern United States, and in blue over here we have Europe. And look at the Southeastern United States. In this area, we have some values that do overlap with the negative range in the Eastern Asia, but we also have a lot of Europe environments represented there. And we do have some environments in here that are not represented in either. But that doesn't mean necessarily that the species cannot make it to those environments, because look at the distribution in Europe and look at here, even though those environments, for example, here were not represented on the native ranges, the species have been reported on those environments. Because sometimes invasive species are constrained in their area, in the native ranges, in the native areas, and once they expand it to a new area, they can explore new environments and they are able to survive in those environments. And that's why I want to talk about prevention and what we can do in order to stay vigilant and proactive against this invader, because as we have seen so far, the species have been expanding very rapidly across the North American landscape. And that's what I mentioned in the last bit of my presentation today is what we can do, we all can do in order to prevent and control these invasive species. So the first thing is like the more for preventing and controlling the spread is monitoring all tree elms that we can, and because that's a key component on detecting the invasion in an early stage and be able to do something about it in order to prevent them to be keeping moving forward. One thing that is very important is to check equipment, cars, firewood that is being moved from areas that are already infected to new places, because the cocoons or the larvae of these species can actually use those places to uh, be moved around the landscape in those way. And this is a picture that I want to share from Autumn and collaborators in 2005 of different cocoons of this species actually being on the mirrors of the car and in the wheel of the car. Every single arrow there is a cocoon. So it is very important that we check every place and equipment and things that we're going to be moving around from invasive areas that are already infected to new locations. Another thing that we can do is to manage this invasion, and because the invasion is not going to kill the tree. There's a few things that, and it's very new in North America. There are not too many things that have been developed yet, but I'm going to mention a few things. And here, first we can, if the invasion is small and the population is small still, and the trees that we have infected are small as well, we can manually remove the larvae and drop them in a bucket with soapy water. We can prune the branches that are heavily infected. There are also some insecticides that have been proved to be useful, and it can be used for larger trees and severe infestation. And those insecticides can be systematic, like dinoterofuran, that have been proved to be very efficacious in removing these species, but also it can be by contact with insecticide and soap or horticultural oils. And we can also be very proactive on the timing that we apply those insecticides, because we have to make sure that the larvae are active at the moment of applying them. And also, there have been some biological control of these species reported mostly in Europe and in Asia, like this fungus that I'm represented here, that have been proved to reduce the populations. And also there are several species of parasitoid wasps and predatory stink bugs that also eat the larvae, but those are mostly on the invaded ranges on Europe and their native population in Asia. We don't know a lot about the biological controls here in North America yet. So with this, I just want to mention that we all have a role in here in preventing, controlling, and mitigating this invasive, and this one and others. And the first thing that we should all do is report it. If we have an insect that looks like a sawfly, like a elm zigzag sawfly, we can take a picture and report it, or we can try to collect it and report it, and we can report it to local cooperative extension officers or forestry departments. They will know what to do about it. We can also report it to early detection and distribution and mapping systems. This is a great resource for compiling all that information. We can also use IMAP invasive. That's also a good resource. And another one that is pretty good is iNaturalist. Like, there are a lot of research grade pictures and observations in here in this platform. Actually, if you remember at the beginning of my talk, I mentioned that this specific invader was first reported in North America through iNaturalist. So it is a very important platform to also be reporting what we found. And we all have a role in that. On this, like, there are not enough scientists to be walking around and monitoring all these pests. So the role of the community and everyone in general is very important for this. So with that, I just want to acknowledge and thank you, my collaborators in the KUENM group. They were the colleagues that helped me develop this research that I show you today. I also want to share, like thank the Invasive Species Collaborative here at Virginia Tech and some funding sources like NSF. And I want to thank you all for being here on my talk. And with that, I will be happy to take any questions.
Thank you so much, Dr. Nuñez-Penichet. Yeah, if you want to give a virtual round of applause, now's the time. That was a great talk. Thank you so much for sharing all that information, especially how management and how to report and things like that. So I do have all the audience muted. So if you have a question, you can either raise your hand or drop it into the chat and I'll read it. And so someone dropped the message in the chat. I'll go ahead. It's John Polo. And they said, does the sawfly select preferentially among different elm hosts? Siberian and lacebark/Chinese elm are from Asia, but have been somewhat invasive in North America.
Yeah, that's a great question, John. No, it looks like they don't have any preference for a specific almost species. They like any almost species. And that's why, for example, in our models, we decided to group the whole genus and model the presence of the whole genus as a one single unit because they use as the host plant every almost species. And till later last year, we thought that that was the only host that they were using, and now we know that they are using another species as well of the Umasia family as the potential host. But yeah, they don't care about that. Mhm.
Great question. Thank you for answering that. Does anybody else have a question? I'm trying to look at the hands to see if anyone has their hand raised. Just raise your hand, I'll unmute you, or drop your question in the chat. We have a comment in the chat. I'm just going to read. Thank you for your presentation. Kudos on a job well done by Michael Manchester. Thank you, Michael. Well, I guess while we wait for everybody else, I'm going to ask a question. So I thought it was really interesting to see that all of the cocoons or I guess, did you use the term cocoon? You can
Yeah.
tell I'm not an insect person, sorry. But they were laid on things other than the elm leaves. So they'll lay them on other surfaces and what then the larvae will crawl to the elm tree. Yeah?
The cocoons, yeah, thank you for the question because I forgot to mention that specifically. But yeah, the cocoons, when the larvae are ready to pupate, they can move. They can do it on the leaf. They will do it on the bottom surface of the leaf, but they can also move down the tree and do it on the trunk or doing in other surfaces then close and next to the elm trees. So they can even do it on the ground and have the cocoon there specifically like on the ground and other surfaces is more common when they're ready for overwinter. So they're going to stay in that stage for for a longer period of time. Um, and after the cocoon, like the pupa period is gone and we have the adults emerging, they fly back to the elm trees and lay the eggs on the edges of the serrations.
Okay, so the eggs are always on the elm leaves. Okay.
Yes, yes.
Okay.
The eggs are always on the elm leaves because they want kind of like give the larvae the opportunity of to start eating as soon as they hatch out of the eggs.
Okay, interesting. Great. All
Yeah.
right. Um, Well, I don't see anyone with their hand up or any more questions. Dr. Nuñez-Penichet, do you have anything else you'd like to share with us? I saw that you have a paper in review, so please let me know when that gets published, and I'll make sure everybody here gets that in our newsletter.
Yeah, I would be happy to do that. Thank you for the opportunity. I very much appreciate everyone's present, and it has been great. Yeah, it has been great to be able to share a little bit more about these invasive species and what we can do about it.
Great. Well, thank you so much. And thank you to everyone who came. I did get kicked off of this video halfway through, so I might not have the entire video, unfortunately, entire recording, but I'll put what I have up on YouTube. So keep an eye out for that for any of you that missed part of it. And thank you again so much, Dr. Nunes Pinochet, and we hope to see you again soon. Bye.
Thank you.